The bad news is that sometimes even single mutations can have a very profound effect on fitness. And now I'm going to give you 2 slides on virulence, so how mutations can make viruses good or bad for our kind of health. The first example is West Nile. So West Nile, this is a tree of West Nile. I'll explain what it is in a second. The Australian version of West Nile is called Kunjin, which is there. West Nile actually was first described in Uganda in the 1930s, and I'll explain what this tree means in a second. Very famously, West Nile was described in New York City in the summer of 1999, a very big epidemic of West Nile. And so it starts off in New York City, in the East Coast here, then it spreads over a number of years across the US, okay? As it spreads, it infects many bird species, particularly the American crow. This crow, it really suffers badly with West Nile. So there's been a massive die-off of crows in the US because of not in my garden, I should say, but in most other places, a massive die-off of crows because of West Nile. So it's a very serious thing. So myself and a chap called Aaron Brought from California and Davis were interested in why West Nile is such a virulent beast in crows. In other species, it's really not. So these other West Nile outbreaks in history, some of them have been very low virulence, very few bird deaths. But American crows drop like flies like a mixed my species. So why is that? So I looked at the genome of West Nile to try and find any mutations that might correlate with virulence. And I found one that's in what's called a helicase, one of the replication proteins, amino acid 249, this particular gene. And the reason why it was very interesting was because every time there was a high virulent West Nile outbreak somewhere in the world, the same amino acid change was seen at this position as a proline. So Egypt, Eastern Europe in the 1990s, and the US ones, very nasty, all had proline. Conversely, cases where globally the West Nile was not very virulent had other amino acids, particularly the blue one, the threonine. So red was bad, blue was not so bad. So what Aaron has this kind of population of crows he can work on. He's going to go to crow hell for this, but he then infects these crows with different strains. So here, and this is what he finds, actually very interesting. So here, this is a very virulent strain of West Nile. This is the crows surviving. This is time. You can see this virus is very nasty, and the crows die off very quickly. And by 7 days in, they've all gone, all dead. He then takes a benign strain, more benign strain, and it's this one, Kenya, wherever that is. Okay, it gets this one here. And you can see here, this is the blue strain, has the blue amino acid. So the red one has, so this strain here has the proline, this one here has the threonine, the blue. See, this virus here, a lot less virulent, a lot less nasty. What he then does, he takes the virulent strain, and by reverse genetics, at that one change, that one amino acid site, he puts in the blue mutation, the threonine, and look what happens. Okay, it attenuates the virus. He then takes the benign strain and puts in the red, the nasty mutation, and it's a high virulence virus. So here, one mutation has completely shaped the virulence of that virus. It's actually very, very interesting. The second example I want to put in is one of the great pieces of Australian science. I've got some new data for you on this. It's Mixomavirus. Now, I'm sure you know much more than me that Australia had a remarkable problem with rabbits, which still has actually. So the first, if I'm correct, the European rabbit was introduced into Australia around Victoria here in 1850. They then spread, 24 rabbits introduced. The rabbits then spread across Australia like this over the next 100 years. By 1950, there may have been 1 billion rabbits in Australia. 1 billion, I just can't believe that number. Okay, 1 billion rabbits. So there's a big, and obviously they're eating crops, do lots of damage. So a very big problem for farmers. So then there was a commission in Australia to ask what to do to control this. And Frank Fenner, a very eminent Australian virologist, said, why don't we introduce some kind of biological control, this thing called Mixomavirus, a South American DNA virus. And this virus, it worked incredibly well, okay? And I should also say it worked so well, the British, following the Australians as we normally do, the British did the same thing in Britain in 1953. They took Mixomavirus as a natural biological control of rabbits. And what happens is a classic story in the evolution of virulence. So here, the virus, this is time, this going this way. This is the virulence. So these are grades of virulence. So grade 1 virulence Mixomaviruses are the nastiest one. The rabbits suffer very badly, and nearly all of them die. Grade 5 is more benign, okay, here. And you can see what happened in both Australia and England. The virus starts off extremely virulent. They're all grade 1 virus, 1950, 1953 here. And then virulence declines, so these lower virulent ones appear here, and then it kind of sets itself at an intermediate level. And that happened both in Australia and in Britain. Now, Frank is a great scientist, now retired. He's in his 90s now, I think. But a colleague of mine, Peter Kerr, has his old freezer from CSIRO. And Peter found in this freezer Frank Fenner's original viruses that he used in Australia. And we've also got a few British ones. So what we're now doing is sequencing the complete genomes of these classic viruses to see what's causing this change of virulence. In particular, what you've done, you've played the evolutionary tape twice. Once in Australia, once in Britain. Have the same mutations occurred in the populations? And the answer is no, it's actually extraordinarily complicated. These are British strains. We're doing this right now, I should say. It's not finished. These are British strains. These are Australian ones. You can't really see it, but the ones in red, it's very hard to see. They're high virulence ones. And basically, they pop up all over the tree. And there is no single mutation that makes this virus benign or not benign. There are many mutations do it. It's a very complicated explanation for virulence. So watch this. We're finishing this off now. Okay. So I've now given you some basic rules of how RNA viruses evolve and emerge. Now, what I want to do is give you two case studies quite briefly. The first is flu. So here are just some very simple figures of more death rates per 100,000 of various childhood diseases in the US over time. You can see we have measles, typhoid, scarlet fever, etc. There was a general decline in mortality due to these childhood infections as our health systems got better over the 20th century. And I want to put on top of that graph, two other diseases, flu and TB. So flu and TB have gone down, but they are still much, much higher causes of death. This extraordinary spike here is the 1918 pandemic, where it had a death rate approaching 600 deaths per 100,000. It's a remarkable death rate. Today in the US, it's still thought about 36,000 people die of influenza every year. Okay, so remember that number, you'll use it in a second. Globally, hard to say, maybe 500,000. So flu is a virus we know a lot about. And over the last 100 years or so, we've had a number of pandemics that we've described very carefully at the genetic level. So the most famous of all was 1918, which the numbers debated, it's between 20 and 50, maybe more than 50 million people worldwide. I'm going to give you some US numbers here. US, over 500,000 people died. It's actually the single biggest death toll in history of any disease in one epidemic. What's interesting is not only was it very virulent, but it hit a very strange age distribution. It hit very strange age groups. So this is a little graph here. This is aged by death rate. Normally in flu, you have what's called a U-shaped distribution. Okay, so it's the very young and the very old. So the most mortality. In 1918, the letter we have is W. So you get a spike in deaths in young adults. And the reason why that is debated. And it's found globally. So it's not just the First World War and soldiers. It's actually a different thing. 1918, 1957, Asian flu, US death toll 70,000. 1968 was bad globally, not really in the US, no more than background mortality. And of course, swine flu, 2009 virus. The CDC, God bless them, estimate 12,000 people may have died of swine flu. Others make it a bit higher. Maybe 60 million people in the US infected. So remember, if that's true, this is a very low number. Even if it's 30 or 40,000, it's not much worse than a standard seasonal flu pandemic. It's not a big mortality cause at all. So now I'm going to give you a very kind of technical slide of the host range of flu. Okay, so here it is. So the natural reservoir of influenza viruses are wild water birds. I'm sure you can see these are mallard ducks, okay? So things like mallard ducks are the natural reservoir of flu, and they contain most of those combinations. And these duck viruses then spill over into other animals. So tigers can get them, whales, camels. Crucially for us, pigs get them, and poultry, so chickens and turkeys get them. And then the pig viruses and the poultry viruses can then infect humans. And it's a big debate, where do we get it from most? Is it birds or is it pigs? To cut a long story short, for many years we thought birds, and we kind of forgot about pigs, and we get swine flu. So they're both equally important. As I told you already, it takes 30 mutations to go from a bird virus to one that works in humans. Horses also get flu, and in the year 2000, the flu virus found a new host species the first time, and that's dogs. The virus jumped from horses to dogs the first time, canine flu. It's quite a big problem in the US in the last 10 years. Now, how it jumped from horses to dogs is not exactly as the hour implies. It was greyhounds in Florida were fed horse meat and the virus was going. So now if you go to dog kennels in the US, you find dogs that they don't really suffer that bad. It's like a bad cough, but it's there in the population. Okay, so I've been involved with lots of projects to understand how flu evolved. I'm going to summarize it very quick because I haven't really got time. But all our analysis has told us is that we can explain the kind of global ecology of flu. Before I show you how it works, one more quick slide. Flu has a very distinctive seasonality. Here's a heat map of flu incidence, so how common flu is, ranked by latitudes. This is north. This is Iceland. Okay, here's New Zealand. Okay, and you can see in the northern hemisphere, flu is a winter disease. Not much in the summer. Winter, not much in the summer. In the southern hemisphere, where we are now, obviously, it's exactly the same thing, but the seasons are offset by 6 months. So in Australasia, around here, it's your winter, which is our summer. I'm sure you know that. What's less well known is in tropical regions, okay, which I've loosely drawn between these two lines here, flu is less seasonal and more continual. You have more continual flu transmission. So remember that fact. So what our analysis has told us then is we think there's a kind of single global reservoir for flu, okay? And it's called what we call a sink source model. And that single global reservoir is most likely, just by default, to be in a tropical region because they have more continual transmission. You have more flu being passed on. And we thought originally it was probably the whole tropics. We now think it's one particular region, and that's East and Southeast Asia. So, to give you one example in a bit more detail, I've been interested in how flu spreads in the US, where I live. And the classic model is that flu comes in on the west coast in California or so, and then moves like a wave across to the east coast over the winter. So we tried to test that. And what we did is we had various sampling. I'll explain the numbers in a second. We have various sampling locations in the US. And I've ranked them here by how west they are. So the westerly place is Portland, Oregon, up there. Most easterly is New York City. This is their longitude. And we sampled viruses from those populations. And the season we looked at was the 2006-2007 season. Actually, not a very good season to look at because it had both H1N1 and H3N2 co-circulating. And then we found out what strain they had of H1N1, H3N2. And the H1N1 strain, or branches, are in capital uppercase. The H3N2 strain, not showing here, are lowercase. So these little numbers here are the strains of H3N2 and H1N1 they have in the population. All I want you to see is it doesn't spread from west to east. If it did, this is the time. Okay, this is October. This is kind of April. If it's going from west to east, you see it going that way. It doesn't. It's all over the place. In fact, what we think goes on is that flu follows what's called a gravity model. So it follows how people move. And people move between, so think of the airline hubs. You have big hubs and small hubs. Flu first goes between the big hubs. Okay, so it goes from LAX to JFK. Then it diffuses around smaller hubs. And it's called a gravity model. And flu follows that. Now, of course, that's seasonal flu, as they call it. Since last year, we've also had this pandemic, H1N1 09, or swine flu. And I'm going to show you how that spread. Okay. Now, just to remind you, this virus has a very complicated history, and I'll try and explain it. But essentially, it's this little figure here is like a road map, like a metro map of the history of this virus. You can kind of ignore it, but where the colours cross, that's a reassortment. That's the virus jumbling its genes up. Very briefly, it emerged when two pig viruses exchanged their genes. One of those pig viruses in the past had a mix of human, pig, and bird genes. And they're called triple reassortment viruses. And that happened about 10 years ago. And those triple reassortment viruses later on, sometime in the last few years, mixed again with the pig strain, and that's the one that infected us. Now, that's well known. What's really interesting is this, though. This is a paper that the CDC published at the same time as they first published this virus. Over the last few years in the US, a number of people have picked up swine virus. It's not the same one that's got going, but very similar. They're triple reassortant viruses. And it hasn't spread any further than one, but they're all spillovers. So these are people, this is their age. They all went to avoid pig fairs. They went to pig fairs, handled pigs, pig farmers. They got flu from the pig, but it didn't spread further. The big question is, why did these not get going and this one did? I don't know the answer. And there you go. So how's this virus then spread? So what I've done with some colleagues, particularly John Romba from Edinburgh, is do evolutionary studies on this particular virus to see its origin and spread. And very quickly, this is a tree of the the swine flu virus. All you need to see are the colours are places. They're very mixed up. This virus is spreading very, very easily around the world. This little graph here, this is a probability distribution of when we think the virus first emerged. And our estimates are, we think it first appeared in humans in the first 7 or 8 weeks of 2009. So January or February, early February, that's when it first appeared in humans. On these data, the most likely place of origin is Mexico. That may change in bigger data. It then spread very, very rapidly around the world. The good news for us is that even today, the virus hasn't made many amino acid changes. It's a very young virus. So the vaccines we have, the vaccine we made last year, still works today. The virus hasn't really developed mutations that make it evade vaccines. So the last 5 minutes, 2 minutes in fact, I'll very briefly do a second disease called dengue. So dengue is unlike flu, it's a vector-borne virus. It's passed on by the same mosquito that infects humans. And I put it into this talk here because it's encroaching into... So basically, because it's vector-borne, its distribution is set by temperature, by where the mosquito can live. And it's encroaching into northern Queensland. And it's normally nasty, it's normally kind of a pain, but not particularly serious. There are some serious forms, though, particularly which can kill kids very frequently. And there are 4 strains of dengue, or 4 types, dengue 1, 2, 3, 4. And like yellow fever, I spent a number of years trying to work out where this virus comes from, how it got into humans. And here's lots of trees, of the 4 dengue strains, 1, 2, 3, 4. And all I want you to see, very briefly, is this kind of, like yellow fever, the time scale, and the fact that in red are some viruses that are found in other primates. So dengue, like HIV, is a non-human primate virus. It's a monkey virus, and it's jumped into humans, at least 4 times. 1 for each of the 4 dengue serotypes. Now, dengue 2 and dengue 4 we have most data for, so we know there's best, but all 4 of them, it would have jumped 4 times, from primates, non-human primates, into humans. And we know the time scale. And it's just like yellow fever. This is a virus that the slave trade, which is terrible for many reasons, the slave trade moved around the world. It's a slave-associated virus. Now, there's a vaccine for flu. There's as yet, there's not a vaccine for dengue. And that's a real problem, because to make a dengue vaccine, there are a few being trialled right now, in fact. To make a dengue vaccine, you need to protect against all 4 strains simultaneously. It's actually quite hard to do. And many people like me think the vaccine we're going to get is not going to work that well. It'll do something, but it'll be imperfect. It won't give you complete coverage, because 4 strains in a vaccine is a difficult thing to do. And so, because it won't work very well, the virus can escape vaccine coverage. We are going to select, by having not complete vaccination, we're going to put selection pressure on the virus to evolve. And there's lots of nice theory, Andrew Reed does very nice theory work showing that if you have an imperfect vaccine, that selection process can result in viruses that are more virulent or less virulent than ones you have. It can change how severe the infection is. So therefore, it's very important to try and predict how the viruses are going to evolve post-vaccination. And I'm going to do this very quickly, but basically what we're doing is going to Vietnam and studying this virus in a kind of micro environment to see how the virus evolves on a kind of micro scale. And that will allow us to predict how it might then evolve given vaccination pressure. And just all I'm going to do very quickly is to see that in Vietnam, what we've seen is, I don't know, go to this slide instead. Basically in Vietnam, we have found a strain called the Asian 1 genotype. And that strain is outcompeting, it's replacing the previous strain that we have. It's called the American Asian genotype. So here's Vietnam. And all we have here is time. This is the percentage of strains in one of those two genotypes. See, it starts off here as the kind of grey, Asian America, and 2003, this strain takes over. In this micro case, one strain is now spread, in fact, not I think micro, all over Southeast Asia, one strain is spread, has replaced the other. Now, why it's important is this strain also is associated with worse clinical disease. So here's viral load in those different strains, and the Asian American one, this is in kids, you have higher viremia, higher viral load, and if more virus, you have worse disease. So this is a more virulent virus. And therefore, it's very important to watch the spread of this virus. Okay, because it could get into Australia. It's a nasty one, and we can see it happening there in the population. This is Ho Chi Minh City, and dengue is like it spreads according to the gravity one, so it spreads by kind of hubs, by density of people, but it's a very slow spreading thing, which is good for us. Okay, so if it does escape from vaccination, we have time to react. It's a slow spreading thing. So our new techniques allow us to actually predict and watch, well, not predict, but watch how quickly these things spread. Okay, so now I'll just wrap up the last two slides here. So I've told you how viruses have evolved. What about the future? Well, it doesn't take a rocket scientist to work out that we are going to get more infections in the future. This is really obvious. Animals carry an enormous number of pathogens, viruses, bacteria, fungi, whatever, that can jump into humans. The way we live today, so we cut down forests, we have changing farming, this is Shanghai, big cities, international travel I showed you already. They are just fuels for viruses. They fuel the engine of viral emergence and evolution. So how do we kind of prevent this? Now, some people say, well, let's try and predict what's going to emerge. Let me tell you, just forget that, okay? I've given you a few generalities. So vector-borne viruses are less likely to emerge, etc. Very simple things. But you can't predict what's going to emerge. It sounds good for the press, but I don't believe it. And I also don't think that going out there in nature and just surveying things is going to be the answer. Because there's just millions of things out there. And that doesn't tell you whether it's actually going to cause disease. To me, the key thing is kind of obvious, is that we need very good surveillance and respond globally. The reason why we need very good surveillance is things like this. This is a map of, sorry, it's not very clear, this is a map of global air traffic, okay, just by intensity. The lighter the colour, the more flights there are. Look at that. This is North America, global economic waster. Here's Australia, and you can see the links to Southeast Asia. This is the kind of problem we face in the modern world. It's amazing connectedness. So we need to respond very quickly. But responding is better than trying to survey things, predict things. And finally, so that sounds kind of gloomy, but I'm actually very optimistic that we can actually do a good job here, because modern molecular biology has also given us some amazingly powerful techniques. So I'm going to leave you with this, okay? And it's a very broad, bold, in fact, it's an extraordinarily bold claim, if not a stupid claim, that what's called metagenomics, which is just mass sequencing, can save the planet. And I'll give you an example of what I mean. It's a virus that I was involved in intimately, and you may have heard of this story. It's the virus that causes a very serious disease of bees called colony collapse disorder. So the bees basically, they get a neurological disorder and they leave the hive and disappear. No one knows where. And this was a massive, massive thing, because this is honeybees. If the honeybees disappear, we are in big, big trouble. Okay, so what caused it? And what we did, we used this metagenomic approach. We basically take hives that have no CCD, that are healthy, and those that are diseased, and we sequence every piece of DNA and RNA in those hives. So mainly we sequence B, but we also sequence all the pathogens they have. And we get a big list of all the bugs these hives have. Then we look for which bugs differ between them, and which ones are only found in the diseased hives. We found one that's called IAPV, Israeli acute paralysis virus. It's not actually from Israel, but there you go. And that is that's a smoking gun. That's the most likely candidate for the cause of this disease. In fact, so my colleagues, this is somebody I worked with, Dan Cox Foster and Ian Lipkin in Colombia. We now can show this virus really does make the bees leave the hive and die. So what I think we should do for the future, we should be bold, is we should go out there and every human pathogen we know, okay, every bacterium, every virus, every fungus, we should have a global survey of their diversity to understand how they evolve. Now that sounds very bold, but the Broad Institute at MIT predicts that by the end of the year, $1 will buy you a whole bacterial genome, okay? or a few hundred viral genomes for that $1. It's a remarkably cheap thing to do now. So I think we have the power, technology here, to really understand how these pathogens spread. So that's basically, now obviously I've summarized lots of people through this. I haven't really got time to thank them all. They're there, they've done a phenomenal job. I'm just kind of like the cheerleader for what their work they've done, but I'll leave it there. Thank you.